My name is Kyla Holt and I'm Head of Volunteering and Alumni Programmes at the University of York. It's my great pleasure to welcome you to this event on illuminating Alzheimer's disease. This evening, hosting our event is Mr. Peter Burley, one of our longest serving and most dedicated volunteers for the University of York. Peter, can I invite you to take over?
Hi, thank you very much, Kyla. Yes, I'm Peter Burley. I chair the London Alumni Committee, and the London Alumni branch is the largest globally of many branches. I graduated from York in history in 1970, and I have to say that my continuing relationship with the university has been a source of great satisfaction. Well, our speaker this evening is Dr. Steven Quinn, and he studied at St Andrews and Oxford universities starting in 2004. His academic career progressed to his current appointment in the Department of Biophysics at York in 2017 in the Biological Nanosystems group. We're beginning to get to the edge of my comprehension here. He works at the interface between physics and biology, and any attempt by me to describe his research in more detail is way beyond my pay grade, so I'm just hoping he can speak for himself there. What I can say is the list of research papers and conferences attributed to him is impressive. I have a personal interest in this topic because my mother-in-law died of Alzheimer's, and my wife Jenny worked for the Alzheimer's Society for several years. And I noticed that Dr. Quinn's most recent career entry is work presented to the Alzheimer's Research UK's conference in March this year. And Alzheimer's Research UK is the UK's leading charity dedicated to researching the causes, diagnosis, treatment and cure for Alzheimer's. And it was set up in 1992 to address the lack of any campaigning body for this sort of academic and medical research. And it has since been behind some of the biggest breakthroughs in dementia research. Well, that's really enough of me. It gives me great pleasure now to hand over to Steven Quinn to speak to us on Illuminating Alzheimer's, shining light on developments in research. Thank you.
Thank you so much, Peter. So I'll just share my screen with everyone, and hopefully you can see this as well as my fancy new laser pointer that I managed to get my hands on earlier this week. So as Peter said, I am part of the Biological Nanosystems group at the University of York, which is part of the wider Physics of Life network. And I'm here today to really tell you that the field of single molecule biophysics is genuinely at the forefront of modern science because it's allowing the very building blocks of human life, so DNA and RNA and proteins, to be viewed and imaged and analyzed. But perhaps more importantly, and maybe even most importantly, for the first time, single molecule bioimaging is allowing us to understand the very building blocks of human disease. And in this talk, I want to showcase how we use single molecule bioimaging approaches to unveil the molecular basis of Alzheimer's disease. And of course, today is World Alzheimer Day, so it's perfectly timed for this talk. So what Alzheimer's disease, what is it? Well, we often say that dementia is the umbrella term for symptoms such as loss of memory, symptoms such as the loss of your ability to critically think and interpret. And of all of the dementias, Alzheimer's is the most abundant, constituting around 60 to 80% of cases. There are, of course, other forms of dementia, including Parkinson's disease, Huntington's, frontotemporal dementia, vascular dementia, Lewy body dementia, and mixed combinations of dementia. But as I say, the key point here is that Alzheimer's disease is by far the most common and the most abundant. And in terms of clinical symptoms related to Alzheimer's disease specifically, we're talking about memory loss, confusion, frustration, disorientation, changes to your cognition, and ultimately personality changes. Now, Alzheimer's Research UK estimate that right now there are around 850,000 people in the UK living with dementia. And of course, that really means that there's over 500,000 people right now in the UK living with Alzheimer's disease. Globally, the number of people living with dementia is immense. We expect that number to increase by around 204% by 2050. And of course, that is a huge economic burden for society, because along with the patients, of course, suffering, there are huge economic costs associated with healthcare. So Alzheimer's disease really is a major global concern for both patients and families and carers alike. And I do encourage you to check out the Alzheimer's Research UK Statistics Hub, where you can really get more of a flavour for the true impact of Alzheimer's disease. But basically, in Alzheimer's disease, brain matter disappears over the course of time. So on the top here, you see a healthy brain, and in the bottom panel, you see a diseased brain. And the clear difference between the two brains is a clear loss of material. So brain cells die over time, and that again leads to death of the patient ultimately. If we look at the mortality rates associated with, for example, stroke, shown in the green curve here, heart disease, shown in the blue curve, and lung cancer, shown in the yellow curve, you can see that certainly the mortality rates associated with stroke and heart disease generally are decreasing over time. Same with stroke. Lung cancer has remained relatively steady. But notice the red curve here. This is the one associated with dementia and, of course, Alzheimer's disease, which is progressively increasing. And again, the number of people living with Alzheimer's disease is expected to increase. That means that the economic costs associated with Alzheimer's disease will inevitably increase. And right now, without an effective treatment, unfortunately, these death rates will continue to increase. So Alzheimer's disease is a major, major problem that we scientists must address. In terms of the biochemical signatures, we think that the first biochemical signatures of Alzheimer's disease appear around 15 years before the first symptoms arise. And there's two signatures, so-called senile plaques, which are made up of beta amyloid proteins, and neurofibrillary tangles, which are made up of tau proteins. So these are the two major hallmarks inside an Alzheimer's disease brain. The beta amyloid plaques are made from the beta amyloid protein, and that in turn is formed by the cleavage, or the snippage, if you like, of a much larger protein called the amyloid precursor protein. So the amyloid precursor protein interacts with basically little pairs of scissors, and we get release of the beta amyloid fragment. And this fragment is a protein. It can fold and twist and bend into a variety of different shapes, but it can also misfold. And when it misfolds, it can cluster together to form these senile plaques. And again, the senile plaques are a characteristic hallmark of an advanced Alzheimer's brain. In other words, if you take an advanced Alzheimer's brain or a brain slice, you will observe an abundance of senile plaques composed of the beta amyloid protein. So these biochemical signatures, we think, begin to appear long before the clinical symptoms first arise. And one of the major unmet challenges facing Alzheimer's disease is to identify people in this early disease phase when the beta amyloid clusters are beginning to form, so that not only we can better understand Alzheimer's disease, but we can intervene before the disease progresses clinically. In other words, before the clinical symptoms begin to arise. So if I just recap, here we have a single healthy cell. The neurofibrillary tangles composed of the tau protein in an advanced Alzheimer patient are generally found inside the cells. The amyloid plaques, on the other hand, are located outside the cells, and both, we think, are extremely toxic. In the context of this talk, I'm going to focus on the amyloid clusters. And again, the amyloid clusters, or the amyloid plaques, are formed through the accumulation of this beta amyloid protein. And notice its shape here. It's an L-like shape with what we call an alpha helical conformation. Notice this helix shape within the L shape. So the beta amyloid protein, again, is formed through the cleavage, the snippage, if you like, of the amyloid precursor protein. So the amyloid precursor protein interacts with two pairs of scissors called beta and gamma secretes. And this is the green fragment the beta amyloid protein that's ultimately released. These proteins can then accumulate into, again, these large amyloid plaques. And here's an electron microscopy image of a plaque that we grew inside the laboratory. You can see that it's several hundreds of nanometers in size, and you can also see that it's very heterogeneous. In other words, there are areas that are less dense than others. There are areas that contain more proteins than others. So this is just a really big, nasty mix of proteins with no real defined shape or structure. So accumulation of beta amyloid is a major hallmark of Alzheimer's disease, and therefore understanding plaque growth and plaque formation is absolutely critical to better understand and therefore treat the disease. So this begs the question, well, if we have the amyloid precursor protein and we get release of this toxic beta amyloid fragment, can't we just stop it from forming in the first place? And so this question, if you like, led to a series of other questions, and one of those included, does the amyloid actually have a normal biological function? In other words, beta amyloid is implicated in Alzheimer's disease. Why do we have this protein in our brains in the first place? So a lot of research went into this particular question because, as I said, if it was perhaps hypothesized that if we blocked the formation of the beta amyloid protein, we could block disease pathogenesis. But the results from this particular question, I think, were quite fascinating. A number of papers suggested, first of all, that the beta amyloid protein could bind to other proteins. So it appeared to have some kind of function, and in particular, it could bind to proteins in the mitochondria of cells. And this is the area of the cells associated with energy production. So there's some links beginning to appear between the beta amyloid protein and energy production. And we also know that it can cross the blood-brain barrier. So in other words, beta amyloid seems also to be implicated in the transport of proteins throughout the body. But perhaps the most interesting of all the papers published in the last 10 or so years was this one published by Robert Moyer's lab. And as the title of the paper suggests, the amyloid beta protein is now suspected to be an antimicrobial peptide or an antimicrobial protein. In other words, we suspect the beta amyloid protein, though constitutes Alzheimer's disease, is also part of the immune system. And what they found was really quite startling. It had very similar properties to the general antimicrobial proteins found in our immune system. It was active against 7 common types of bacteria, including E. coli. And importantly, it had a striking resemblance in terms of its properties, and in many cases outperformed the most abundant antimicrobial protein in our body, LL37. So taking all of this evidence together, there's really good suggestions that the beta amyloid protein actually is essential for our normal biological function. But of course, it's clearly implicated in Alzheimer's disease. So amyloid is part of the immune system. So what are the key open questions? As I've mentioned, we get plaque formation through the accumulation of the beta amyloid protein. But what we don't yet understand are what specific cells are involved during this toxic protein cluster formation, and when do they become involved? And specifically, what are the first of all the cells to be involved? And what follows when a cell perhaps interacts with a beta amyloid protein cluster, and why? And as a consequence, we don't fully understand how the brain circuits actually fail. And underpinning all of this, we still really don't understand fully what nanoscopic mechanisms are involved. And this is where my lab comes in. We are specifically interested in understanding the very building blocks of the protein clustering process, and importantly, how these individual and very distinct protein clusters cause toxicity. And one of the hypotheses that we're working towards is that the amyloid protein clusters effectively punch holes within the cells. And that can lead, we think, to the leakage of vital cellular contents that leads to the cell ultimately dying. We also don't know whether or not these processes begin at birth. So I've said that these biochemical signatures appear to begin around 15 years before the first clinical symptoms arise, but really we don't know what triggers this protein clustering in the first place. And the ultimate goal, the ultimate open question is, can we achieve sensitive, early stage diagnostics and effective treatments? And both of these questions are questions which we and my colleagues are also working on in York. We're trying to develop rapid and sensitive and multiplexed blood testing technologies to try and identify people in the earliest possible stages, such that we can more effectively and more efficiently search for the next generation of therapeutics. So with that being said, my colleagues in York span biology and chemistry, physics, and the social sciences, trying to understand Alzheimer's disease across multiple length and timescales. From the very earliest building blocks of beta amyloid protein assembly, which can take place over several nanoseconds, that's a thousandth of a millionth of a second, right the way through to entire populations of people and how these populations interact. So my colleagues in social policy are interested in population studies. My colleagues in biology are interested in understanding the global dynamics of the organs involved in Alzheimer's disease, of course, the brain being a key player. We're also interested in the behaviors of cells and different types of cells in response to stress, and in particular, in response to these toxic protein clusters. And the physics department are largely involved with the generation of tools, typically optical tools, for studying protein structure. And why are we interested to study protein structure? Well, protein structure is related ultimately to protein function. And in my lab, which is based largely in the science part, we're interested in understanding the building blocks of Alzheimer's disease on the nanoscale. And very recently, we've installed a new piece of instrumentation funded by the EPSRC, the Engineering and Physical Sciences Research Council, to understand Alzheimer's disease on a timescale of picoseconds. So that's a thousandth of a nanosecond, and a nanosecond is a thousandth of a millionth of a second. So we're really trying to interrogate Alzheimer's disease at the very, very earliest stages. So if I go back to the beta amyloid protein, these proteins are produced and they ultimately form these large plaques, which we find inside the brain. But in order to get to the plaque-like stage, they have to cluster together, first of all, into small clusters that we term oligomers. And these oligomers, as they progressively get longer and larger, lead to the formation of long stringy-like objects that we call fibrils. And these fibrils, as they fold and twist and bend and really manifest themselves around each other, lead ultimately to the formation of the plaques. And whilst early research, in fact, the earliest research into Alzheimer's disease focused specifically on the fibrils and plaques, recent evidence now implicates the small oligomers, the earliest assemblies, if you like, as being orders of magnitude more harmful. In other words, we believe that the damage is done in an Alzheimer's brain at the very early stages of protein assembly. And so a number of questions have arisen as a consequence. And the main question is, how do these oligomers form? And importantly, which oligomer, if any, is the most toxic? And ultimately, how can we stop them from growing? So as I've mentioned, a number of tools and techniques have been developed largely by the physicists to understand the structure and ultimately the function of specifically the fibrils and plaques. So electron microscopy and a number of biological stains have been developed for that purpose. And in our lab, we're using single molecule bioimaging technologies to really address open questions in this part of the aggregation spectrum. So what is single molecule bioimaging? Well, you can think of single molecule bioimaging as an extension to normal microscopy. So we know when we look down the barrel of a microscope, we can very easily distinguish between, for example, live or immobilized organisms, human hairs, biological cells, even bacteria. In other words, we can identify objects that are typically millimeters in size down to objects that are typically a few micrometers in size. A micrometer is a thousandth of a millimeter. But we cannot, using conventional microscopy, identify clusters of viruses. We cannot differentiate multiple viruses in a cluster. We certainly can't differentiate using optical microscopy certain proteins in a cluster. They're simply too small, and we certainly cannot image single molecules. And that's because of this term up here, Abbe's diffraction limit. Optical microscopes are really, really good at allowing you to identify and distinguish between single clusters when those clusters are relatively far apart, so greater than 200 nanometers in terms of their spatial position, but they're really not so good at differentiating between objects on the nanoscale. And this is where single molecule bioimaging comes in, because the trick here is to attach a small fluorescent dye molecule to your protein of interest. In this case, that allows optical microscopy to visualize the protein clusters or the viral clusters or the clusters of molecules. Now, in the context of our work, we attach highlight fluor 555, shown in the red star, to our beta amyloid proteins. So this is a little fluorescent dye molecule that absorbs strongly in the green parts of the electromagnetic spectrum. In other words, it absorbs green light very well, and it strongly emits red light. And that red light we can capture using our optical microscopy techniques to obtain images such as the one shown on the right-hand side. So essentially, I take my fluorescently tagged beta amyloid proteins, I allow them to cluster together. I can change the environmental conditions. I can switch, for example, the salt concentration in a solution. I can change the acidity of the solution. I can change things like concentration. I've got very good control over the environmental conditions of the beta amyloid protein in the test tube. And when I've formed clusters, I can deposit those clusters onto a glass microscope slide that sits on top of our microscope. I can then inject laser light from this box into the microscope slide. That then stimulates the fluorescence emission, the emission of the red light from my sample, and I can collect this on a camera that we cool to about -80 degrees Celsius. And again, we're left with images such as the one shown in the right-hand side, clearly defined spots that exist against a dark background. And what you're observing here are single clusters of the beta amyloid protein, and the blue dark background is the microscope slide. So we can now, using single molecule bioimaging in combination with the attachment of a fluorescent dye molecule, clearly resolve objects that are much smaller than the Abbe diffraction limit, 200 nanometers. So how are we doing this in a little bit more of the physicsy detail? Well, we're using a technique called total internal reflection fluorescence microscopy. And this is allowing us to visualize not just single protein clusters and single proteins, but hundreds of those single proteins and hundreds of those single oligomers in parallel and in real time. So what we do is we take our laser beam, and rather than simply injecting it into the sample, we deliberately shine it off of the glass microscope slide such that it reflects rather than refracts into the sample. And we do this because Maxwell, famous physicist, tells us that at the point of total internal reflection, when this beam becomes reflected, we generate a secondary beam of light that penetrates into the sample. And that secondary beam of light penetrates only around 200 nanometers into the sample, which basically means that we only get light emission from molecules, from fluorescent molecules, which are sufficiently close enough, i.e. attached to the microscope slide. That allows me to minimize any background noise and obtain what we call in physics a good signal to noise ratio. So I can minimize my background counts and I can maximize my signal counts. The fluorescence is generated and we collect it from below using an optical lens, and we pass that fluorescence on to our cooled camera. And again, we get an image that looks something like this, where each spot corresponds to a single oligomer. So not only can we collect images, we can collect timestamps, we can monitor these spots in real time, we can monitor how they might evolve over time, and importantly, we can extract the intensity of a single cluster and monitor the intensity over time. So what you see on the right-hand side here is a 3D representation of the image on the left, where each peak corresponds to the presence of a single protein cluster. And just notice qualitatively that some of the spots are brighter than others, and that's indicative of some of the clusters essentially being larger than others. Now, the way to interpret these microscopy images is much in the same way that you might interpret a satellite image. So here we have a satellite image collected from the NASA Earth Observatory of the United States. And what you can clearly see is a series of bright spots or bright regions against a dark background. And of course, we know where Austin is, we know where Atlanta is, we know where New York City is, and you can see that these areas are the brightest of the bunch. Now, in Austin, in Atlanta, in New York City, we of course have a number of people. We have a number of apartment blocks, a number of street lamps, a huge number of office buildings. And what I'm getting at here is that the brightness of the city or the brightness of the town is a good indicator of the population. So if you look closely near Chicago, you can see again various little towns appearing, and in the background you can also see much less bright spots corresponding to villages. So brightness is really an excellent indicator of population. The more office blocks we have, the more apartment blocks we have with their lights switched on, the more street lamps we have, is a good indicator of the number of people residing in those areas. So let's imagine we have a city in the United States of America, and let's imagine we ask everyone to turn their lights on inside their office block, inside their apartment block at the same time. And now let's imagine I look at that brightness of the spot over time. And assuming all of the light bulbs are functioning correctly and everyone keeps their lights on when I ask them to, then over time, the brightness of the city will remain relatively constant or unchanged. But then if I ask people to randomly switch off their lights, so to progressively run around their office blocks or their houses and turn off the light bulbs one by one, then as they do this, the brightness of the city will become progressively dimmer and dimmer. And as each light bulb is turned off, the city brightness will ultimately decrease in a stepwise manner, until such a point where all the lights in the city have been turned off and the city brightness turns to zero. So using this trick of monitoring the brightness of a spot in this example, I can estimate to a very good degree of accuracy the number of light bulbs or the number of lamps that might exist within a particular city. So the number of turning off events that I observe is an excellent indicator of the number of light bulbs present in the area of interest. And this is the trick that we use to investigate the building blocks of Alzheimer's disease, because we take our fluorescent images of the preformed protein clusters or of the single proteins that exist on the microscope slide, and we monitor their intensity over time. And in much the same way as your light bulbs in your house or your office have a lifetime, in some cases several years, several months, the fluorescent dye molecules also have a lifetime, and they can turn off quite rapidly after a few seconds, especially when a high power of green light is instant upon them. So in other words, we can deliberately ask these fluorescent dye molecules to turn off one by one. And so what you see in the middle here is a 3D representation of fluorescent protein clusters. This is in real time, and you can monitor that these peaks gradually disappear. If I zoom into a single peak, perhaps you can see that the turning off occurs in a stepwise like manner. So in other words, if I have, for example, a protein cluster that contains 4 individual proteins, and each protein has a fluorescent tag attached to it, then if I monitor the brightness as a function of time under my conditions of high excitation power or high green light power, I will monitor a number of turning off events. And the number of turning off events, or so-called photo-bleaching steps that I observe, will directly correspond to the number of single proteins within my cluster. So in the example I've got here, I see 4 turning off events, and that tells me I've got 4 single dye molecules inside the cluster of interest, and that corresponds to 4 single proteins. So the key point from this slide and the take-home message is that the number of photo-bleaching steps corresponds to the number of single proteins, or monomers as we call them, inside a single oligomer. And so using this trick of stepwise photo-bleaching, we can identify single proteins. So this would be a single protein with a single dye attached to it. In other words, we observe 1 step. We can identify the earliest stage of protein self-assembly, in other words, a dimer, a protein cluster with 2 single proteins in it. And this is a trace that would show 2 steps. We can identify those with 3 and 4 and 5 steps, such as the one shown on the bottom left-hand side. So a protein cluster containing 5 single proteins should display 5 turning off events, and a hexamer, a protein cluster with 6 single proteins in it, should display 6 photo-bleaching steps, like the graph shown on the bottom right. Now, why do we want to do this? Well, we think that a dimer has very different levels of toxicity to a pentamer, and a pentamer has very different levels of toxicity to a hexamer. So by interrogating our overall sample and understanding which types of species we have, we can more effectively understand which are the most toxic, and therefore perhaps the most relevant for inhibitors. So we can get unique access to monomer to oligomer aggregation using this technique. We can even go as far as to identifying protein clusters that contain around 32 photo-bleaching steps. We think in the literature at the moment, the record is around 10, but as I say, we think we can beat that by a factor of 3 and identify 30 tumors. Essentially, the more and more fluorescent dye molecules you have, the more noisy your data becomes, and it becomes even more difficult to quantify particularly large structures. So single molecule bioimaging is really great for understanding the earliest stages of protein self-assembly, but less so for understanding the larger clusters. Brightness is also a really good indicator, as I've mentioned. So the overall initial brightness will scale with the number of single proteins within the cluster. So we can also use brightness as an indicator. And in this particular sample, we can see the majority of species based on the brightness were monomers. So these were species that contained a single step. There was some levels of dimers, around 30%. So those species displaying 2 photo-bleaching steps. And we even had a few trimers, those which displayed 3 photo-bleaching steps. So photo-bleaching plus brightness are 2 optical signatures or optical fingerprints of Alzheimer's disease on the nanoscale. And it's become a bit of a tradition now that when we collect data like this for the first time, that we stop everything that we're doing and we immediately grab the cameras and take a photo. So this is myself and Dr. Alex Payne-Dwyer, who spent an enormous amount of time building the microscope and collecting the single molecule stepwise photo-bleaching data for the first time. Now, why is this important? Well, we can access heterogeneity within a sample. We can identify how many monomers there are relative to dimers and how many dimers there are relative to trimers, for example. So here's an example of an aggregation system where at time 0 minutes, the majority of species displayed 1 protein or 1 peptide per oligomer, but there was some evidence of dimers with a few trimers. And under the environmental conditions that we used, you can see the evolution of this histogram over time. So in other words, we see an increase in the number of dimers and trimers and pentamers and tetramers, even after 15 minutes. And that progresses even further at time 1 hour. So now the number of monomers in the system has vastly reduced, and the system, the solution, is swamped with oligomers. But importantly, after 1 hour and in the presence of an inhibitor, you can see that this histogram is very, very similar to that obtained at time 0. So this is now telling us that we can block the aggregation using putative inhibitors. So single molecule bioimaging technology is not only giving us a new understanding of what particular species might exist within a sample, but it's also leading towards more effective and efficient drug screening, as we can monitor what happens in the presence of a potential aggregate blocker. So just to summarize, we have a situation in Alzheimer's disease where these single proteins progress into fibrils and plaques via oligomers. A number of microscopy-based techniques and a number of biological stains have been developed to identify the fibrils and plaques. But where there's a real gap in our knowledge regarding how the monomers progress into oligomers, we're using single molecule bioimaging techniques, which is essentially utilizing fluorescently labeled beta amyloid proteins as a means to provide unique insights into oligomer formation. For those of you who are interested in this work, we have now published this paper in the journal Methods. This is an open access paper, so it's free to view. It's titled Amyloid Beta Oligomerization Monitored by Single Molecule Stepwise Photobleaching. And our hope is that the biologists, the physicists, and the chemists of this world will take our tools and techniques and more effectively and efficiently screen for the next generation of inhibitors. This work would not be possible without the combination of chemistry and physics and biology, and therefore the combination of chemists, physicists, and biologists. And we're really blessed at York that we have a really great team of interdisciplinary scientists working on this project. So I have to thank Alex Payne-Dwyer for building the microscope. Lara Dresser was heavily involved in collecting much of the data that you observed today, along with Patrick Hunter. Jamie Howard did a lot of work on the protein purification side of things. Ji-Yun Lee, who is now working in Edinburgh, did a lot of work on figuring out how to immobilize the proteins onto a microscope slide so that we could study them. Thanks to also Professor Mark Leake, who heads the Physics of Life Research Group in York, and also to Gareth Evans, who we're working with in trying to understand which of these protein clusters are most toxic. I also need to thank Alzheimer's Research UK, who have supported all of our work. Quite simply, this work would not be possible without their funding. And I need to also thank the EPSRC specifically for funding Lara in this work. And of course, thank you for your attention. I'm now very happy to take any questions. Thank you.
Right. Well, thank you very much indeed for that. Absolutely fascinating. I'm not sure I grasped every subtle nuance of all technical data, but I am going to ask you a question straight away, which I'm reasonably confident I will understand the answer. And that is, you mentioned that today is World Alzheimer's Day. Can you just tell us when that started, why it was set up, and what it aims to achieve, apart from us listening to talks like yours on this particular date?
So Alzheimer's Day is part of Alzheimer's Month. And the main goal here is to ultimately raise awareness. If I put it into some kind of perspective, the amount of funding that's available specifically for Alzheimer's disease research is about equivalent to 1 mile of motorway per year. Compare that number with the funding that's associated with cancer and heart disease, HIV, and stroke. The amount of money that's available for Alzheimer's disease research is utterly minuscule. Now, that's not to say it's not effective, the money that is there, but ultimately we need to raise awareness of dementia-based research, but also the implications of dementia and Alzheimer's disease generally for the people who live with Alzheimer's disease and dementia, and for those who care for people with Alzheimer's disease and dementia. Because as I mentioned, the cost is huge, not just to the person with the symptoms, but also to the costs, economic costs involved, both to families and to governments. So Alzheimer's Day is really about raising awareness and really, demonstrating that this is a disease that we understand. I mentioned at the start that we have some open questions, but by and large, we understand what's going on. We understand these protein clusters exist, and therefore a therapeutic is in sight. And we really just need to push towards that finishing line now.
Okay, thank you very much. Now, I've got two questions from Wolfgang. And the first is, I'll give you them both because I think they may be linked. The first one is, how can you ensure that each protein has its own fluorescent dye molecule? And then he goes on to ask, can you ensure that the inhibitor is not destroying the protein function?
So to answer the first question, how do we ensure that each protein has its own fluorescent dye molecule? So we can employ a number of tricks and techniques during the purification process of these things. We can try to separate free fluorescent dye molecules from proteins, but also labeled proteins from unlabeled proteins. That works to some degree, but really to identify whether or not every single protein has been labeled, we can perform a technique known as absorption-based spectroscopy. So really what that means is quantifying the amount of dye that we have in our sample versus quantifying the amount of protein that we have in our sample. And it seems based on our data that we have a 1-to-1 labeling ratio. In other words, the amount of dye present in our final sample matches very well the amount of final protein. How can we ensure that the inhibitor is not destroying the protein function? It's a great question. Many inhibitors, many putative inhibitors, can interfere with proteins. In other words, they can simply just change the structure of the protein, and that leads to it not being able to aggregate. But as I mentioned, the beta amyloid protein we think is part of the immune system, so it's needed inside the body. So we can't do that with an inhibitor. What we can do, though, is test the amyloid function using a variety of different assays. And in this particular case, the inhibitor is not interfering with the function of a single amyloid protein. But great question.
Right, thank you. Now I've got two questions that I again suspect they're related, from Sandy Divers. And she asks, how long will it be until the process of research is available to Alzheimer's patients directly, and/or are human clinical trials in process already, please? And then linked to that, additionally, are you able to direct different procedures more specifically to the previously acknowledged Alzheimer umbrella definitions? E.g., will the different Alzheimer definitions require different processes? And I should be very grateful if you could address those.
So to address the point, how long will it be until the process of research is available to Alzheimer patients directly? So a lot of this work has been used with my colleagues and others, as I've mentioned, for the development of a rapid blood test. So moving away a little bit from inhibitors and understanding the disease, we now have the tools and techniques in place at York to study single protein oligomers, and importantly, to detect single protein oligomers at really, really, really low concentrations. Now, many of the conventional blood tests can't do that. They just don't have the sensitivity or the specificity, and our techniques do. So what we're trying to do at the moment is build upon preliminary data to build a blood test that we can then commercialize and put into clinical trials. And we hope to do that within the next 2 years. This is all entirely dependent on funding. We've got many applications at the moment pending. We're very hopeful that this work will be funded. We hope that it will be funded. And if it is, then we have a commitment to our funders to get these tools and technologies out there within 2 years. In terms of the inhibitor work, so we're if you like, at the very, very early stages of the development of an inhibitor. So we're in the test tube. And really to develop an inhibitor that will ultimately be used in humans, there's a number of different steps that have to be fulfilled, a number of requirements, a number of boxes that have to be ticked. So we're at the very early stages of that. And I would guess that from the test tube to a clinical drug would be around 8 to 10 years. But the good news is that technologies are evolving all the time. Technologies are getting better and faster. Well, as we know, because of the coronavirus pandemic, and we're now able to screen for inhibitors much more effectively than we were able to several years ago. So these timelines are constantly evolving over time. Are we able to direct the procedures more specifically to other forms of dementia? Yes, the answer to that is absolutely. We are using these tools and techniques to investigate the building blocks of Parkinson's disease. So we're looking at protein aggregation, but not in the context of the beta amyloid protein, but in the context of the alpha-synuclein protein, which seems to be a major hallmark of Parkinson's. And in motor neuron disease, we now believe that the clinical symptoms are related to the buildup of a protein called TDP43, and we're using our tools and technologies again here to understand the building blocks of motor neuron disease. So absolutely, that's exactly where we want to go.
Thank you. So that's a very encouraging response. Now let's move on to Andrew Porrer, and he asks, how can you disrupt or destroy the toxic stages of this process without stopping the protein fulfilling a useful function elsewhere in the body?
And this is the $1 million question ultimately. So we need the protein. We can't get rid of it. We can't just simply stop it from forming. But equally, these protein clusters do seem to be toxic. So the key goal here is to block the toxic formation. Now, that's not to say that the overabundance of the single proteins could also happen, and we could get even more protein clustering after the let's say the drug or the inhibitor has gone in. It's a great question. We don't know that we can't disrupt or destroy the protein function, but I think I would take my chances with disrupting the normal protein function versus the protein clustering, which seems, as I mentioned, to be orders of magnitude more harmful.
Okay, I've got a question from Sue Lister, who's asking this as a non-scientist. I think some of the question you may have covered, but I think it's such an important area. If you are repeating yourself, I think it's worth doing it. Right, she says, what is the safe way to block the growth of dementia, and when will this be available to the NHS? But before this, how and when may you be able to test for the pre-dementia symptoms?
So right now, as things stand, there have been over 300, believe it or not, drug trials aiming to identify an inhibitor, a blocker, if you like, for these structures. Not one has been successful in a clinical trial. And we think it's because the drugs are targeting the wrong structures. We think that the drugs are targeting the fibrils and the plaques and stopping the fibrils and the plaques from growing even further. They're not targeting the oligomers. And recent developments have seen at least in the test tube and in animal models, the development of inhibitors, which do seem to be blocking the oligomer. So I think there's promise there. When will this be available to the NHS? Again, it takes time for these inhibitors to be fully validated. But I think the timescale that we're working on is on the year's timescale. How and when may we be able to test for the pre-dementia symptoms? Well, I would go one step further. We want to really test for the biochemical signatures long before the symptoms actually arise. Once it gets to the point of having symptoms, we think that the plaques have formed. So really what we want to be able to do is identify someone who has no symptoms, but who is showing biochemical signatures. That comes back to my previous answer of we hope to have a blood test technology in about 2 years. We also hope to use the technologies of the blood test to detect proteins within the urine. So not just a blood test, but a non-invasive test, if you like, that might be able to identify whether or not you have more oligomers relative to proteins in the body, and therefore whether you are exhibiting the earliest biochemical signatures of Alzheimer's disease, such that we're in a position, a far better position, to help slow those symptoms down. Because there are drugs available to help slow the symptoms, but there's not a drug yet to completely block the Alzheimer pathogenesis.
All right, thank you. Now I've got a very different question from Ben Wagner, who asks on a personal level for you, what made you first pick up Alzheimer's as an area to research?
So my grandfather had dementia. I'm sure he had Alzheimer's disease, though it was never really clinically diagnosed. And part of the problem is in large part because there is no diagnostic test for Alzheimer's disease. But in a sense, his mind dies. So names, dates, places, his entire scrapbook, his entire mind dissipated. And for me, that was really quite eye-opening, and even more so because there really was no treatment at the time. So Alzheimer's disease, I think, much like Peter, it hits home. In Oxford, I studied cancer-based research at MIT in the US. I studied cancer-based research, but albeit on new tools and technologies. And now that I've studied a number of different diseases using a number of different tools and techniques, we're now in a position here in York to more effectively pinpoint and target the earliest stages of Alzheimer's disease. So I think just to broadly answer your question, it was a personal relationship with my grandfather that probably directed me into the area of research that I'm involved with now.
All right, thank you. Now I've got another question from Wolfgang, and this question makes me suspect you may know him. And he asks, would there be a benefit to measure the absolute concentration of those proteins, brackets, regardless of the, I think it might, he says clustering, I don't know if it's clustering stage, within a probe without single molecule detection? If so, are there standard techniques at hand? He says hyperpolarized 129XE might be there as a tool. At York, there is also a great group working on this. I think your answer might perhaps unpack a bit more of what he's asked there, if you can.
So the answer is yes, there is a benefit to measuring the absolute concentration of proteins, because it's the overexpression, the abundance of these proteins that seems to trigger the clustering process. So in other words, the more beta amyloid proteins we get, the higher chance of clustering. More so than that, there are different lengths of the beta amyloid protein, and measuring the ratio between these various lengths is thought to be an excellent indicator of Alzheimer's pathogenesis. So specifically, to answer your question, there's a beta amyloid protein that's 42 amino acids long. There's another one that's 40 amino acids long. There's another one that's about 16 amino acids long. And yes, measuring the absolute concentration of those proteins in the blood, in the urine, could be an excellent indicator of Alzheimer's disease. The trouble, though, is that there is not a standard tool for measuring picomolar concentrations of proteins, so really, really tiny amounts of proteins. The best technique is a technique called ELISA that we have, but the sensitivity isn't quite there for the concentrations that we think are in the brain, sub-nanomolar, at the very early stages of disease pathogenesis.
All right, thank you. Now I've got a question from an anonymous attendee, and they ask, do you think environmental factors could slow down the development of clusters? Yes,
much of our work in the test tube does show that environmental factors can influence the growth of these objects. So specifically, the salt concentration, the local salt concentration that's surrounding the proteins influences and certainly speeds up the protein clustering. Switching the pH is important. So the more acidic the environment is, the faster the clustering becomes. Now, quite how those environmental factors we can change is a question that I don't have the answer to. Is it related to diet? We just don't know. But certainly the local environment of the protein does heavily influence the development of the clusters. Now, that's not to say that they won't form if the environment wasn't changed or wasn't altered in some way, but there does seem to be a link there between the growth rates.
Right. I'm afraid we're going to have to wind this up very shortly. I've got two more questions, so perhaps a quick answer to both of them, if you could, and then we'll keep our time limit. From Linda, she asks, following your research, do you think pharmaceutical companies will become more interested in developing potential preventative drugs than they have been lately? And actually, in a sense linked to that is Andrew Porter's come back with another question. At what stage is the most damage done, and can it be reversed rather than just stopped?
So to answer the first question, I think the answer is yes. So for example, Johnson & Johnson, I know, are particularly interested in developing potential preventatives for not just Alzheimer's disease, but dementia in general. They're also, I know, really interested in the technologies. So in the sensing technologies, they want to send out blood test technologies to GPs so that they can more effectively know who's in the early stages of disease pathogenesis. So I think there's two prongs there. First, yes, they're interested in preventative drugs, but they're also interested in the technologies. Now, at what stage is the most damage done? So that's the question that we're trying to really address. We don't know whether it's the trimers or the tetramers that are more toxic than the dimers, or vice versa. What we do know, though, is from our preliminary work that some of these oligomers can punch holes within the cells. And when they punch holes within the cells or the contents of the cells can be leaked to the environment. And that, I'm afraid, cannot be reversed, at least in the test tube.
Right. Well, thank you very much indeed. I say it's been a very, very stimulating evening. It's wonderful to have this world-breaking research showcased for an alumni forum. And I really do hope we can arrange some more of these. I've really enjoyed hosting it. I've really enjoyed listening to you. And thank you for speaking to us. Thank you to Kyla for arranging all of this and her team that's made it all possible. And thank you to everybody who's logged on and listened to the talk. Thank you again.
Thank you on behalf of the whole university to Peter for hosting this evening's event for us, and to Steve, not just for giving up his time as well this evening to speak to us today, but for everything that he's doing. For the fantastic research and for the encouraging news. Thank you very much, Steve.